I told you would be a large part of your test in understanding the arrows. And what I mean by the arrows is when you map out the overall equation of, let's start with photosynthesis. Okay, on the left side of the arrow in a chemical equation, you would put glucose, you would put carbon dioxide and you would put water. And on the right side of the arrow, you would put glucose and oxygen. And I don't know, in my 10th grade science classes, it looks like you just magically take carbon dioxide and water and just poof, it just turns into sugar and oxygen. But we now understand that arrow going from our base raw materials to the sugar and the oxygen, that arrow is a very complicated arrow with lots and lots of steps. So we're going to review those arrows real quick. We will try to clear up some of the confusion here. This will definitely be like essay questions and many questions on a test. So the first question I wanted to answer is what arrows? So I thought this was a neat little diagram because it shows both photosynthesis and respiration and kind of highlights the fact that they're essentially opposite, chemically speaking. With just if we were just talking about writing a chemical formula, the chemical formulas end up being pretty much exactly opposite of each other. So this is both photosynthesis and respiration on one diagram here, and all they did was flip the heads of the arrows back and forth to show you that you go back and forth between one or the other. But the arrow in photosynthesis involves different steps than the arrow in respiration. So I want to make sure you understand that even though you start with these raw materials in respiration and end up with these finished products in respiration, and you do exactly the opposite in photosynthesis, they're not really exactly opposite as far as the nitty-gritty detail goes. When we actually dissect these arrows and look at what the process is, different things happen during the process of converting the raw materials into the finished products, depending on which reaction you're talking about, photosynthesis or respiration. So I'm going to start at photosynthesis, and I'm going to quickly go through what happens at the arrows. This is by no means a complete step-by-step, every little detail summary, but it gives you at least some idea, and it'll be helpful for studying. So step one of photosynthesis is light reactions. They look like this. And during the light reactions, we start with photosystem 2. Photosystem 2 splits water. High energy electron from water gets pumped through this electron transport chain, which takes H+ ions and shoves them into the thylakoid. Also, we get some H+ from broken water that ends up in the thylakoid. So you end up with this thylakoid that's very full of these hydrogen ions. Okay. So the next part of the light reactions was the electron transport chain. Then we do photosystem 1, where we take more sunlight energy and we re-energize the electron, and then we dump it off into this last step where we build a high energy electron carrier. The other molecule that's an output, so this is one output of the light reactions, this high energy electron carrier here. The other output of the light reactions is going to be ATP, because what you do with all these H pluses that you've loaded into the thylakoid is you allow them to flow out through ATP synthase, which mechanically cranks the ATP synthase, makes it go round and round, and that mechanical energy gets converted to bond energy. That's what ATP synthase does. So through that whole process, through just the light reactions of photosynthesis, your output ends up being NADPH and ATP. You're going to use those high energy molecules and you're going to dump them to the Calvin cycle. So the outputs of the light reactions are the inputs into your next step, which is the Calvin cycle. Speaking of the next step, all right, there we go. The next step of photosynthesis are the dark reactions, also called the light independent reactions or the Calvin cycle. Dark reactions is kind of a misnomer because they don't necessarily happen in the dark, but they don't need light to proceed. All they need is those high energy molecules from the light reactions. So here's a little diagram of the of what's going on with the dark reactions. This is where CO2 comes in. CO2 is on the left side of our arrow of the overall equation of photosynthesis. CO2 is an input into the dark reactions, as well as here's the ATP from the light reactions, here's the NADPH from the light reactions, here's more ATP from the light reactions. You need all those to drive this cycle, to keep this cycle going. What you do is you take CO2, you add it to this molecule called RUBP, which is a waste from a previous trip around the cycle. So this gets added to CO2, incoming CO2 gas. We process this molecule. Okay, we build it up until we got our final product down here. The final product of the dark reactions of photosynthesis is glucose. And if you remember, the overall goal of photosynthesis is to make glucose. So we are officially done with photosynthesis now. The plant has achieved its goal. It's made glucose. But it's got some waste left over, and that would be RUBP, which is going to get added to CO2 again. Here's my arrow again. And that CO2 added to RUBP is going to allow us to continue this cycle and build more glucose. That's why it's the Kelvin cycle and not just the Kelvin straight line. It's a continuous cycle. So that's step 2 of photosynthesis, and we're all done. We've achieved our goal. We've made glucose. Let's see here. So the next part, I wanted to just review here. So CO2, if you look at the overall equation of photosynthesis again, CO2 went in during the dark reactions. Water went in as a first step to the light reactions. What you got out as waste product was CO2. Actually, that's the main product, is glucose that I just circled. I can't talk anymore. Glucose is the main product that came out of the dark reactions. Oxygen is a waste product that came out of the light reactions of photosynthesis. So, this arrow includes both the light reactions and the dark reactions that I just went over. So that is definitely a complicated little arrow. And again, I didn't cover every single detail, but you're going to be responsible for knowing all the details that we went over in notes when we get there on the test tomorrow. More review of the arrows, because we've only done photosynthesis so far, so we still have respiration. Step 1 of respiration is the Krebs cycle, also known as the citric acid cycle. Those are the same things. Here's a picture of the citric acid cycle. Again, it's a cycle. The waste products from a previous trip around the cycle are going to help start the cycle again. This nasty little molecule at the top here, acetyl-CoA, he is pyruvic acid that has been processed a little bit. To keep things simple, I guess that's how we could describe that. And if you remember pyruvic acid, that acetyl-CoA used to be, pyruvic acid is really just glucose that's been split. So you can think of acetyl-CoA as being a slightly processed glucose. What we're going to do is we're going to take that acetyl-CoA, we're going to add it to oxaloacetate, which is the waste coming from a previous trip around the Krebs cycle. So you're going to add these two together. You get this first carbon intermediate called citrate. That's why it's called the citric acid cycle, because another name for citrate is citric acid. You're going to take citric acid and you're going to break it down in a series of steps that's going to release a bunch of energy, but you don't want that energy to come out spilling out as heat and light. So instead, you lock up that energy in a chemical bond in this molecule, NADH, in this molecule, another NADH. Here's some ATP spilling out. Again, this is because we're breaking the bonds in citrate, releasing energy, and tying it up in all these energy carriers all the way around the cycle. When you're all done, you're back at oxaloacetate again. You're going to add it to more broken up glucose that's coming in at the top of the cycle. You're going to build this large complicated molecule that you're then going to break down in a series of negative delta G reactions to start the whole process over. That's the first step. What you've got when you're all done with the Krebs cycle is you have high energy electron carriers and you have a fair amount of ATP produced. Those are the outputs of the Krebs cycle. Those outputs are going to be fed into the second part of respiration, which is, wait for it, the electron transport chain coupled with chemiosmosis. So that looks like this. What we've got going on here, this is sort of a complicated drawing, but I kind of like this drawing. Here's the inner mitochondrial matrix in the middle here. And what we're doing is running the citric acid cycle in the middle of the mitochondrial matrix. It's pumping out these high energy electron carriers, which we then run through these protein pumps in the membrane. These protein pumps are grabbing H pluses from out here, and this should start to look familiar. H pluses that are out here in this intermembrane space between the inner and the outer membrane of the mitochondria. There's all these H pluses. These fired up proteins here that we just ran a high energy electron through, they act as pumps. They're going to grab that H plus, they're going to cram it inside the mitochondria into the inner matrix. See, there's ones down here that are doing the same thing. So now we load up the inside of this mitochondria with all kinds of hydrogen ions. They're all over the place. Lots and lots of hydrogen ions. Okay. So basically what I've done here is this is all loaded up in the middle. Lots of hydrogen ions. They're all over the place. Look at them. Hydrogen ions don't like to be near each other, like we described when we did chemiosmosis with photosynthesis. These hydrogen ions want to escape from each other. Their only escape route is through ATP synthase, which of course when they go squirting through ATP synthase, they make ATP synthase actually mechanically go round and round. That mechanical energy gets turned into bond energy in ATP, and that's how every living thing on the planet is making ATP, is by cranking around in ATP synthase. And ATP is, again, our basic energy currency for the cell. So at ATP synthase, or ATP, once it's made, can go diffusing out of the cell and drive all of our cell functions that we've been talking about all year so far. So there. And let me see. Oh, again, the complicated arrow. So I wanted to just reiterate one more time that that arrow in the overall equation there for respiration that I just threw up at the bottom, that arrow is a complicated process that involves the Krebs cycle, and the outputs of the Krebs cycle get fed into the electron transport chain, which drives chemiosmosis, which actually ends up making the ATP. So that is that. And that is a caterpillar dog. Your test is coming up here very, very soon. This might be helpful to review before the AP exam as well. I hope it kind of clarified some things. Most of that was items you already had in your notes, but I just want to make sure that I had made every effort to make everything clear. So good luck with your studying, and have a good night.